And and so that we are sort of semi on time and not carving too deeply into Andrew's time, we're really happy to have what I've referred to today as a trifecta of wonderful speakers that are going to walk us through a scientific journey that really does read like a detective story. And in fact, Lauren Aguirre has written a book that features part of this journey, trying to understand the fact that fentanyl not only has obviously devastating effects in terms of lethality, but it also may have some very direct and specific effects on the brain in terms of brain damage. And we have wonderful representation here from the sort of earliest inklings of this in the field with Andrew Kofke, who's been at Penn since 2001 after earlier training and earlier positions, which I won't go through, but is director of anesthesiology and clinical care, and basically laid the preclinical foundation. Then we're going to have Jed Barash talk about the sort of early clinical part of this journey when he began to notice individuals who had a very profound amnestic syndrome after what looked like an opioid overdose that might have involved fentanyl. And then we will finally have Ignacio Badiola tell us a little bit about the clinical trial that's going to be running here at Penn, already underway for the pain patients and potentially for having an OUD component, that will be examining this in a prospective way, looking for people who have documented overdose and looking for the specific kind of brain damage that may have effects above and beyond just plain old reduction of oxygen that can occur with an overdose. So let's get started, Andrew. You have wonderful slides, and we will start this journey now. We're happy to have Lauren Aguirre with us here today, by the way, who is the author of the book, Memory Thief.
Got to get everyone's pictures out of them. Okay, this is going to be the title of this is Pursuit of the Memory Thief, stolen from Lauren's book, but it's describing an association between opioids, fentanyl and its congeners, and acute hippocampal injury.
Do you want to go to the slide full screen?
Yeah. Okay.
Great.
So this describes much of the story from Lauren's book, which you can see here, and there was a podcast that came out on Friday where Lauren was interviewed by the PBS folks, and there's a URL there, so you know that it's online. I'm going to discuss preclinical studies. Jed is going to discuss the clinical discovery. Basically, he called it connecting the dots, and Ignacio is going to review the funded research protocol. So on my part, discussing opioid neurotoxicity, the preclinical studies, and human volunteers. No conflicts of interest. I tried to get companies to fund this, but for some reason they wouldn't fund it. So this starts with seizures. When I was during my critical care fellowship, I was asked to give anesthetics to patients for seizures, and that led to a research line looking at anesthesia interactions with seizures. And as I was doing that when I was at Hershey from 83 to 88, their reports had come out about opioid-induced seizures. So I did some studies looking at, since fentanyl was the new opioid that was out, I looked at fentanyl and other drugs, which you'll see. So it starts with seizures, and then it leads to other issues. So here's example of the relevance to humans, and it's seizure-induced brain damage in humans. So on the right, you see a normal hippocampus, and on the left you see one that came from someone who ate mussels. I don't eat mussels anymore, but the person who described this still consumes them. Jeannie Teitelbaum in Canada. So I described domoic acid because it's an analog to canic acid, which has a lot of similarities to fentanyl, and this is an example of a canic acid analog which produced brain damage in humans. So there may be a link to opioids. So this is canic acid seizures in rats, and this is the seizure activity that you can see here, and it produces brain damage. And this is a light microscopy showing a layer of injury here in the hippocampus, and then electron microscopy showing injury from canic acid. So that's all good. And then, but then whenever you compare the autoradiograms for canic acid, which is, as I say, is a analog for domoate, which produce damage in humans, canate versus fentanyl, there's a baseline autoradiograms. This is deoxyglucose autoradiography, and here it's a striking similarity in the autoradiograms, and they both produce seizures, and they both produce this pattern on autoradiography in rats. So let's get to the work that I've done with rats, and notably, my rats were always physiologically controlled, which is important in these sorts of studies, seizure studies and opioid studies, that there's no hypoxia, there's no hypertension, no hypertension. So these are rats that were, this is actually on a ventilator, lying on the ventilator, with a temperature probe, a femoral blood pressure monitoring, a ventilator, expired gases being monitored, and EEG being monitored. So all the studies that I'm going to describe, and many others which I'm not going to describe, all involved neuromuscular blockade and physiologic control of the animals with appropriate anesthetic. So here's an example of an EEG from alfentanyl, which is a congener of fentanyl induced seizure. And this is the surface EEG, but we also wanted to confirm that there was limbic system depth EEG like seizures, and we saw it on depth EEG and also on cortical EEG in rats. So I was in a laboratory where we did radioactive glucose autoradiography, not deoxyglucose. So it was a shorter snapshot of time. So the rats had seized for, I think, a half an hour or an hour with alfentanyl. And then we did injected the radioactive glucose and then perfusion fixed and so on, and processed them for autoradiography. And the redder it is, the higher the activation, and this is the control. And here, when the alfentanyl treated animals, you can see that the ventral hippocampus was activated with, and it may become, I didn't realize it at the time, but it becomes important that the amygdala was quiet. So we did histopathology in those studies and other studies, finding selective cell loss in the hippocampus. And here's another example on light microscopy. But notably, in a couple of the rats, we had an amygdala infarction. So the amygdala was pretty much wiped out, and here you can see there's some hippocampal injury, and notably the amygdala seems to be an area where the autoradiogram was quite quiet. So I suspect that this is reflecting injury. So then we did some dose response studies, and this is number of animals with normal histology, with control meaning no fentanyl, and then increasing doses of fentanyl with loading dose and maintenance dose gradually increasing. So we saw that there were more and more animals that had brain injury, and then as we got to higher doses, I was surprised that it seemed to get less, and I wish I had gone to one more higher dose to see if it had started to look like here, suggesting that there's an optimal dose. I didn't prove it, but it certainly is suggestive. And then because opioids are associated with hypoxia, and because in surgery, patients who get opioids often have brain ischemia, we looked at a study with if animals that had forebrain ischemia in the context of fentanyl administration. So this is a number of animals here, and then we had a pathologic grade, 0 being normal for the pattern of injury, and 5 being the worst. And this is the just giving fentanyl. And then interestingly, the injury got worse, the ischemic injury. This is not the limbic system. This is the striatum, which is the caudate and the putamen in the rats, and that was that ischemia model. That's where the injury was. So I was actually pretty surprised to see that another area of the brain was made more susceptible to ischemic injury in the context of fentanyl, and I'm not sure the circuitry that supports that, but it was an observation, and it becomes important whenever we're thinking about the cases that we're going to be hearing about later. It's like you say, well, gee, they were hypoxic, and well, maybe they were hypoxic, but if they were in the context of fentanyl, this would suggest that the ischemic injury is just a bit worse if there's fentanyl on board. And then we did studies with Remy Fentanyl, which is a drug that came out subsequently. And this is important because the Remy Fentanyl, you give the exposure, you turn it off, and you can extubate the rats right away, and the Remy Fentanyl is gone. It's a very, very short-acting drug, so there's no issues with ongoing injury after the experiment when the rat's in the cage. And this just shows this is using fluorojade, so that whenever they light up, that means the cells are injured. So moving on to people. Some people studies. This is just a paper. It's not an opioid paper, but it's just a great illustration of an epileptic patient getting ketamine with depth electrodes, and the depth EEGs get very activated while the cortical stays perfectly quiet, and underscoring the importance of thinking about depth EEG subcortical seizures. And indeed, someone reported that with fentanyl. This is an epileptic patient with hippocampal electrodes on one side, and he with I think it was a modest dose of fentanyl, 10 mics per kilo, and the anesthetizing dose would be 100 mics. He produced seizure activity in the hippocampus on this patient. And then another study looking at by Lee Kearse, who's a EEG trained neurologist who also couldn't make up his mind and went and did anesthesia after that. So he's double boarded, and he did an EEG study with it in cardiac surgery patients who were getting very high doses of fentanyl for induction, and he didn't produce seizures, but he produced epileptiform activity. And then because of the seizure activity, we decided to see, I wonder if it's a good drug to give during ECT in patients who have depression. And here we saw the amount of volts, I guess this is volts, millicoulombs required to produce a seizure, comparing, this is session numbers, they get serial sessions, and this is, and here the methotrexate, the amount of electricity that was required came up and plateaued. Remy Fentanyl was much lower electrical stimulus required. And then the duration of the seizure was longer with Remy Fentanyl compared to methotrexate. Indeed, I had one patient who I gave a loading dose of Remy Fentanyl to before the ECT, and he seized, which led to a discussion as to whether the ECT was required or not. They went ahead and did it anyway, but it was just, I don't know. I don't know if that was appropriate or not. Maybe Remy Fentanyl is good for depression all by itself. So these are some monkeys that we gave fentanyl to, and with CMRG PET back in Pittsburgh, and these are the baseline images, and then whenever they did digital subtraction after we added fentanyl, the temporal lobe was shown to have increased rate. It was only 3 monkeys. It wasn't enough for the journal to accept, but it's an abstract form. And then we did some humans intubated. So human volunteers intubated with neuromuscular blockade were then given the FDA-approved high dose of Remy Fentanyl. And this is statistical parametric mapping, which indicates statistically significant areas of activation. And here we see the temporal lobe activated and a little bit in the cingulate. And this is just a summary of that, showing the brain region of interest that activated with CMRG with Remy Fentanyl in humans, and the temporal lobe activated. And I must say, this is the most stressful study I've ever done, putting volunteers to sleep and putting them in a PET scanner. And then we did a study here at Penn using arterial spin labeling CBF and sedative doses of Remy Fentanyl, so they weren't intubated. And here we can see the increasing doses of Remy Fentanyl, and it gets a little bit redder because there's global increase in CBF, and that's just from CO2, because CO2 increases. So then after we correct for CO2 and look at various regions, this is the region of interest divided by global. We see statistically significant effects, and the amygdala goes down, the cingulate goes up, the hippocampus goes down. I'm not sure what to make of these in terms of brain damage, but the point is that the areas of interest in the rodents were affected in the humans. And then as sort of a looking under a rock kind of a thing, we just said, well, I wonder if there's an ApoE effect. And comparing E4 with not E4, we saw the amygdala, E4 had a higher CBF, and the cingulate had lower CBF, and the hippocampus had higher CBF. Again, I'm not sure how to interpret that, but it was an observation that there might be a genomic influence for this phenomenon. So in In summary, then, we have in rodents, seizures clearly produce brain damage. In humans, seizures produce brain damage. In rodents, opioids can produce seizures and hypermetabolism in brain areas which subserve cognitive function. And in humans, opioids produce seizures and hypermetabolism in brain areas subserving cognitive function. In rodents, opioid-induced seizures produce brain damage in brain areas subserving cognitive function. And of course, we don't know in humans because no one will volunteer, but we're going to do, well, you'll hear how we hope to look at this. And then in rodents, canic acid limbic neuroactivation is virtually identical to that produced by mu opioids. And then domoic acid, which is a canic acid analog, was consumed by humans and produced seizure and limbic damage. Cognitive effects of opioids have not been assessed in rodents, but in humans, cognitive deficits occur after surgery, and of course, every surgery involves the use of opioids. So these are the folks that I worked with over the years at Penn State. John Graybill was a technician. Rick Hawkins was my mentor. He was a Krebs-trained neurochemist, so I learned a lot from him. Then in Pittsburgh, Marie Rose was the fantastic technician who solved every problem. Bob Gorman was the pathologist who we did all of the pathologic evaluation with. At West Virginia, the folks who took care of the patients, so I didn't have to stress out over it, was John Barbacci and Alicia Sins. And then at Penn, Eileen Maloney helped with the CBF studies with Pat Blissett, who was a postdoc, and we used John Detry's technology. So I just ended with what I was told at around 1990 when I was an assistant professor. This guy who was big guy in anesthesiology comes up to me and he says, "So you're the one who's going to get us all sued." So on to Dr. Burp, Dr. Jed.
Thanks. Ignacio has my slides, I think, so I'm going to see if you can put them up.
Let me put them up.
Thank you, Andrew. Great foundation.
Appreciate that. All right, so should I just tell you next when to go?
Sure.
Yeah, that'll work.
Okay. Thanks. So thanks for starting us out, Andrew. I'm Jed Barash. I'm a neurologist from Massachusetts, and I'm doing kind of the clinical portion of this talk. Next slide. I have no conflicts of interest to disclose. Next. So this is the twofold objective here. One is to just understand the evolution of an epidemiological investigation, and the second is to learn about the opioid-associated amnestic syndrome, which is the subject of the epidemiological investigation, and also an increasingly common phenomenon that we're seeing now. It's a kind of an emerging condition, and that'll be the kind of the focus of what I talk about today. Next. So basically, it started back in October 2012. It was my second month of being an attending neurologist. I was at Leahy Hospital and Medical Center in Burlington, and we had a 22-year-old guy who came into another local emergency room. He had a history of heroin and polysubstance use. He had injected heroin, woke up, and it had presented to that emergency room within a few hours, experiencing some tingling in his extremities, which turned out to be kind of pressure-related neuropathy, but more pronounced was this anterograde amnesia, which was really dense. His mother had he was transferred to us, and his mother had presented with him and confirmed that it was more than just kind of just general intoxication, that it was, he was basically asking questions over and over again within, seconds, couldn't really retain any new information. And so he wound up getting admitted to our hospital for a workup. The urine toxicology was positive for opiates, which wasn't surprising given what he had told us. And we did a whole workup, including EEG, spinal fluid, infectious serology, hypercoagulability studies, all of which were unrevealing. But what was really striking was his MRI of the brain basically had a complete wipeout of the the hippocampus on both sides bilaterally. It's called restricted diffusion. And next slide. And what you'll see is in the images, you'll see kind of this area here. That's the hippocampus, which is outlined in white, and that's actually the image itself. It's not highlighted in any kind of way, but these are axial images, so they're slices kind of almost like horizontally at the level of the eyes and moving up and down. The last image here, the MIP, is a maximum intensity projection, which is almost like a summary of all the slides and kind of captures the summary of the hippocampal damage that was done. You can see it pretty clearly on both sides here, and it was something that almost looked like a stroke. That's what we would call that like hippocampal ischemia. But what was unusual was that it was so localized to the hippocampus. It wasn't in a clear vascular territory like we would normally see. And I had certainly never seen it before. None of my colleagues had seen it. We weren't sure what to make of it. Next slide. So he had additional cognitive evaluation. Even 4 weeks later, he had moderate to severe impairments in his, his storage, retrieval, and recall. Even at 6 months and 2 years later, he had some, really mild residual deficits related to that. Avi, you go to the next slide. And so, we had done a literature review to see if there were cases that had similar imaging, because I had mentioned, none of us had seen it. There were a few kind of sporadic case reports in the literature associated with lone cocaine use, none of which, by the way, were tested for synthetic opioids, which is, relevant later on when we get to testing for fentanyl. There was a case in Korea of carbon monoxide poisoning. There was a case, I think, out in California in 2014 of a kind of an immunogenic response to an influenza infection that had a similar imaging finding. And there was a case in 2013 of a similar finding on one side, one side of the brain with heroin inhalation in France. That was pretty much all we could find. So we thought about writing it up, and I think like a lot of things, one thing led to another. We got very busy with other things, and we wound up kind of sitting on it, and that was that for the time being. Next slide. So in January of 2015, I'm in clinic, and I have a patient, is a 41-year-old guy who's booked to see me for memory loss. And I didn't have too much information for the visit. He was coming from out of state. He was kind of like a secondary, like a second opinion. But the story was basically he had a history of heroin abuse by inhalation and had reportedly been abstinent for years. He had a history of high blood pressure and otherwise pretty unremarkable medical history. His family had gone away during Thanksgiving weekend, and they called him at some point during the weekend, and he was okay. And then they called him again, and he couldn't figure out like why the kids weren't home. They should have been in school. Again, asking kind of repeated questions. They came home, they found him to be what they called, quote, confused. He went to the emergency room with them the next day. He was pretty hypertensive in the emergency room, and they documented him as being amnestic on bedside testing. His short-term recall was impaired. It was pretty rudimentary, but it was observed. They didn't do a real detailed workup. Like he didn't have a toxicology screen sent. He did have liver function tests that were done. Those were mildly elevated, which we wound up seeing in other cases. But his other diagnostic workup, including an MRI, was reportedly unrevealing. He hadn't saw a local neurologist later on, had an EEG that was, unrevealing. But the memory deficits continued. They were saying like he was having trouble learning rules to a board game for the kids, very like simple stuff he couldn't learn. And in my clinic, when we did a Montreal cognitive assessment, he lost 4 or 5 points for recall, which is, pretty bad for anybody, but never mind a 41-year-old guy. Next slide. So what happened was I wound up getting the disc from them, and I wound up not having a ton of time like I normally do, like was normally the case. And I was like, well, I'll look at it at the end of the day. I always looked at my own images, but I was like, it was read as normal. I'll look at it later. So when I popped up the images, it looked exactly like the same images as that patient back in 2012. It was, strikingly the same thing. I have the same kind of arrangement here where you can see it again, the summary image that shows the damage. And so it was like a flashback there, going back into time. And I thought it was, such a striking finding. And the fact that both of them had a history of opioid use, I was like, well, that's kind of weird. This patient denied using opioids, but again, he had no recall. He couldn't possibly remember at this point, and there was nobody with him. So we weren't going to be able to, validate that one way or another. Next slide. So he wound up getting, we asked him to get an MRA, of the head and the neck, looking at the blood vessels at an outside facility, which was done about 14 weeks from the onset, just to make sure there were no clots or anything else. That was unrevealing. And he was lost to follow-up. I wound up contacting his primary later on, and he had died from cardiac arrest within about a year. I think it was August the following year, so less than a year from the onset of presentation. Next slide. So that was the origins of our investigation. So I spoke with our neuroradiologist, Juan Small, and he had taken an interest in the cases. I had him look at that second case just to make sure I wasn't seeing anything I was onto something. He agreed it was really odd. And we agreed basically to seek other cases through a systematic review of hospital patients there, which we wound up doing. And then through that process, we learned of a third patient that had occurred in November 2014 in our hospital. And then while I was on service, prospectively, another one in November 2015. So we had 4 patients, and we felt like there was enough there that there was some meaning to this. And the other 2 patients also had a history of using opioids. So there was a connection there. We wanted to investigate it further. Next slide. So I wound up contacting the Massachusetts Department of Public Health. I'd worked with the state epidemiologist there, Al DiMaria, previously on research involving prion disease, and so we had an established relationship. And I think he had some trust in what I was telling him. And I wound up describing the cases to him. He had said, okay, let's go ahead and see what we can do. I had actually, I think, come to him after I had 2 cases. I had approached the CDC. The CDC kind of brushed it off as maybe incidental to other things, and Al said, "Why don't you collect more cases?" Which is what we did. We got to the 4, and then he said, "Okay, let's see if we can take a look." There was a doctor by the name of Nick Somerville who was embedded with the Massachusetts Department of Public Health. He was with the Epidemic Intelligence Service in the CDC, so we took an interest in getting him on board, and he was interested because it kind of fit the type of work that they were doing. So we worked together to craft a communication from Massachusetts Department of Public Health through the Board of Registration in Medicine to neurologists, radiologists, and emergency room physicians to report similar cases for medical record review. Next slide. So the cases we were asking them basically was someone from 2012 forward, so basically from the time that we had seen the first case, who had new onset amnesia in the absence of evidence to support a readily apparent cause. In other words, that there wasn't some other obvious cause for the amnesia, and changes consistent with acute and complete ischemia of both hippocampi and MRI at initial assessment. So, and that was kind of the descriptor we used for the finding. Ischemia is essentially like a stroke or lack of oxygen. Of course, at the time, we didn't know what the exact mechanism was, but we used that as the terminology because we felt that that would be the terminology that was most readily familiar to people who would be receiving our communication. It was basically like a common language, and we knew that they would know what to look for if we were describing it that way, and that was how we picked that language. Next slide. So what we did was we asked people who were submitting the cases to give us the information we needed to track down the available clinical data. So that included notes, diagnostic testing, any other kind of data related to substance use. We took that data and we abstracted it into a database where we could kind of compare the patients, and it would allow us to kind of observe any kind of trends that we wanted to identify. Next slide. So the response is pretty striking, actually. We got cases in response to our email. We got cases within minutes. That's not an exaggeration. So people, without knowing what we were talking about, knew what we were talking about, and responded right away. So aside from our 4 initial cases, there were 21 others. So we got 25 patients dating back to 2008. And 14 of those met the definition we established. So we basically, after we reviewed those records, those are the ones we honed in on. 11 of them were retrospective, dating back to 2012. 3 of them were prospective after we released the letter. So they were new cases after February 2016. None of the patients who we received reports on before 2012 met the clinical case definition. Okay, next slide. So 9 of the patients were unconscious at the time they came to medical attention, and of those 5 required intubation. And then of the other 5 patients, basically what happened is family members, friends, or acquaintances basically came upon them after a limited time apart, noticed that they had severe memory loss, and brought them to the emergency room. Next slide. This is kind of a summary table that we used in MMWR, where this was published, describe the group. Essentially, the age range was 19 to 52, is more bimodal in the 20s and the 40s, predominantly male sex, which, younger and male sex is kind of reflective of a population that uses, that is more prone to using substances and illicit substances, substance use disorders. There's a lot of detail here, but the next slide kind of summarizes. So if you go to the next slide, kind of go through that in a little bit of more detail. So there was a history of substance use disorder that was documented in 13 of the 14 patients. The 14th patient tested positive for opiates and cocaine at the time that they presented. So essentially, they all had either some history or of use or positive toxicology. The other patient who tested positive for cocaine also tested positive for opiates and a few other substances. The reason I mention that is just because of we didn't have a case of we didn't include any cases of lone cocaine use because of the idea that it was recognized in prior case reports. So we omitted those. But 13 of the 14 had either a history of opioid use or a positive toxicology for opiates. That was 8 of them tested positive for opiates, and none of them at the time had tested for synthetic opioids like fentanyl. We'll get back to that. There were 2 patients who had negative toxicology results, and one of them reported heroin use in the days before the event, and the other had a history of opioid use, but we couldn't get further details on him. Next slide. And this is just kind of, a summary of kind of what the images looked like, similar to before. The one thing I would point out that I bring this slide up is this patient, for example, had basal ganglia lesions here too. So some of these patients also had lesions in other areas of the brain. It was most commonly like subcortical, like in the brainstem, or basal ganglia, or in the posterior aspects like the cerebellum. And that's kind of like just a representative, but they were all linked by this kind of common hippocampal finding. Next slide. There were no abnormalities on the EEG in the 11 patients who had that. Spinal fluid results were unremarkable. We even tested for carboxyhemoglobin and methemoglobin in a couple of cases that we were able to identify, and that had that testing. That was negative, given the report of a case of carbon monoxide poisoning in Korea. LFTs, the liver function tests, were almost uniformly elevated, and otherwise the workup was unremarkable. Next slide. So the deficits generally improved over time. There was memory loss in one patient at 5, that resolved at about 5 months, and persisting in 2 patients for follow-up of more than a year. A lot of them were lost to follow-up, and we couldn't. It was hard to know. MRI abnormalities typically resolved. We had one at 5 weeks in 2 other patients. There was some volume loss that was noted. One patient had a seizure disorder after the evaluation for the amnestic episode. Next slide. So our conclusions were this was a combination of clinical findings that was rarely seen, and in isolation. The cluster was unusual because of the absence of a readily identifiable etiology, the clustering of in time and space, relatively young patients, and extensive substance use among those people. We were not sure of what the mechanisms were, but we thought further exploration might identify that. Next slide. We made it a reportable disease in May of 2017. So essentially anybody who saw this had to report it to the Department of Public Health. We asked people to basically do a workup that included MRI of the brain, toxicology screening, neurology consult. But the big thing was that we asked people to do advanced laboratory testing for synthetic opioids, because nobody had tested for fentanyl. We were suspicious that fentanyl was the cause. The reason being, fentanyl was increasingly common in Massachusetts from 2012 forward, and that's when these cases started emerging. People had been using, heroin for years and years, but it seemed a little unusual that it would have just emerged at that time. But the timeline for us seemed suggestive to be consistent with fentanyl use. And so over the next year, we wound up collecting some cases from people reporting it. Next slide. So these were 2 publications. One was in Annals of Internal Medicine. This is a group from West Virginia. Was worked on separately, identified a patient there who wound up testing positive for metabolites of fentanyl with this syndrome. And that was kind of the first time we were able to link fentanyl with this syndrome. He had also tested positive for other substances, so there was a little bit of a confounding factor. But we were able to identify 4 cases in Massachusetts, which we reported in the New England Journal in March of 2018. And 2 of those 4 tested positive for fentanyl only. They had extensive workups. And so it became clear to us that fentanyl seemed to be the most likely culprit here. Next slide. And so that led me back to identify this work that Dr. Kofke had presented previously, and we wanted to kind of try to work to connect the endpoints that we were looking at, both kind of preclinical and then kind of clinical and epidemiologic, into a kind of a unifying paper. And we wrote a kind of review that kind of went through kind of the evidence connecting fentanyl and opioids with hippocampal injury. There was a subsequent case actually in Canada the following year of a woman who overdosed on fentanyl patches. These were prescription fentanyl patches. And that was a particularly interesting case because, number one, it was a case that was outside of the United States, number one. And the second was she was using prescription fentanyl. The question had come up previously was, well, why isn't this just a contaminant? You have no idea, these people are using street drugs. Well, this was a case where a woman overdosed with her partner next to her. I think she was found in bed next to him after she had put, I think, 3 fentanyl patches on. And that's what led us to say, you know what, this has been a trend that's going on from 2012 to 2019. So you're talking 7 years, multiple countries. That seems to be less likely consistent with what would be an adulterant or a contaminant. Those are usually kind of isolated, short-lived phenomenon. They're usually not widely spread over many years. So that was kind of the connection we made. Next slide. And we came up with a definition of the syndrome here. So basically, we put together that in 2020. We confirmed cases, probable cases, possible cases. Confirmed cases had a positive toxicology and MRI evidence or CT evidence of the injury. Probable cases had at least a history of known opioid use and imaging evidence, and possible cases would have kind of one of either positive toxicology, a history of opioid use, or imaging evidence of damage. Next slide. These are the people who have kind of worked with consistently over the years on this topic outside of Andrew and Ignacio. Al DiMaria at DPH, Nick Somerville I mentioned, Monroe Butler, who worked with me, and Yuval Zubar, who worked with me at Leahy, along with Juan Small, were critical in identifying the earliest cases. Chun Lim at Beth Israel, a neurologist there, who has seen a number of cases as well. Michael Lev in radiology at MGH, Mark Hout at West Virginia, who helped us with that case that I pointed out earlier. And then 2 ER toxicologists, Mike Ganetsky and Katie Boyle, as well. And that's it.
That's terrific, Jed. We really appreciate it. Do you want to take any questions now, or you want to plunge on through and have Ignacio introduce the prospective study that's going to go on? Would we?
No, you can Ignacio. Let Ignacio go, because I think I ran a little over. So.
Okay, that's fine. Ignacio, we're very happy to have you talk about how these connected dots are leading to the forthcoming clinical study.
Great, thank you. Hold on, let me pull this up. All right, can everyone hear me? Oh, good.
Yes.
So based on the work that Dr. Kofke and Dr. Barash have done over the last few years, we put together a study which was just recently funded in R21, which I'm going to discuss now. Just quickly as a background, chronic pain is a big issue that affects a big proportion of the population. About 20% of the U.S. population suffers from some form of chronic pain. And about 8% of the population suffers from chronic pain of the lower back, which is usually defined as pain that persists beyond 3 months. So obviously a big problem. And until recently, opioids were the cornerstone of treatment for a lot of pain conditions. Obviously, as the opioid epidemic has kind of sprung into full bloom, there's been less prescriptions given by providers with more reliance on other forms of therapies, including things like interventional therapies, physical therapies, cognitive behavioral therapies, and the like. But even then, still about 5 to 8 million Americans use opioids daily. Some have been on them for very prolonged periods of time. For example, in our clinic, we have patients that have been on higher doses of opioids now for 10, 15, 20 years and have been relatively stable. So even though the main gist is to kind of pull patients off opioids, some of these patients still continue to benefit from chronic opioid use. This is just a picture of something that came out a little while ago, last month I think it was, from the CDC, showing that the number of overdose deaths has increased in 2020, no doubt, somewhat from the pandemic. But you can see that the numbers have been steadily rising over the last few years before that as well. So in terms of side effects, opioid medications do have a lot of side effects. These are some of them here, the ones that we typically think of when we prescribe them, and the ones that we tell patients that they're more likely to have would be things like nausea, vomiting, constipation, urinary retention. But there have been obviously things like respiratory depression where people have died from opioids. And the thought is that a lot of these side effects tend to reverse themselves when you stop taking the opioid. And so if you're constipated, you stop taking opioids, the constipation should get better. Another side effect that is not typically included, but that if you ask patients, are memory problems, especially in older patients. And you'll see patients that start taking opioids. We follow up with them, and they may tell you that they have problems remembering where to put their keys. They can't remember things that are on the tip of their tongues, and they forget their train of thought. And temporally, you would think that stopping these opioids would tend to make things better, and a lot of the times they do. But one of the questions we have is, given the previous data that Dr. Kofke and now more recently Dr. Barash have come up with, do any of these individuals, even though we stop prescribing opioids, have we caused some kind of damage, especially in chronic users, to their hippocampus, where we may not see the very severe effects that Dr. Barash has described, but maybe some subtle effects that have gradually worn down and caused subtle memory problems in these individuals. And for the sake of time, I'll skip this, but this just shows that that there is memory side effects associated with acute chronic use. Acute opioid use. And so the question that we had based on the last few years and the last 20 years of Dr. Kofke's work is whether patients who use opioids to treat chronic lower back pain, again, that being one of the most common pain conditions in the United States, especially at higher doses for a prolonged period of time, will they have smaller hippocampal volume on MRI and perform worse on neurocognitive tests of memory compared to chronic back pain patients who do not use opioids. So again, going back to these individuals who have been on these opioids for a long time, their constipation is well controlled. They don't have nausea and vomiting. They've been on these same medications for 20 years, 15, 20 years. Has there been subtle issues with their hippocampus and memory from their chronic opioid use? And so the study that we designed that was just funded, and we're hopefully going to be recruiting our first patient in a week or two, is a cross-sectional study in patients that have chronic lower back pain. So we've made the group a little bit more homogeneous by excluding other forms of chronic pain, which may have some neurocognitive issues associated with them as well. Our cases are going to be individuals on high-dose opioids, which we define as greater than 90 morphine equivalents per day. This is what the CDC has classified as higher dose. We have a lot of individuals who are on significantly higher doses and have been for 15 or 20 years. Some patients on 200, 300, 400 milliequivalents of morphine. Our controls are going to be individuals that have chronic lower back pain, which we define as longer than 2 years. But again, we're going to search for individuals in our clinic who have had chronic back pain longer than that, and have instead used non-opioids and no other medicines really to treat their pain chronically. So we do have a fair amount of patients who are not really interested in medications, and they've instead treated their back pain conservatively with things such as interventional therapies, and even things like physical therapy. We're going to have 35 patients in each one of these groups, and the study is slated to take about 2 years to complete. So the two outcomes that we're going to be looking at are hippocampal volume using magnetic resonance imaging, and cognitive function using a computerized neurocognitive battery, which is made up of 14 tests and was developed here at the University of Pennsylvania by Dr. Ruben Gur, one of the neuropsychologists, and measures multiple domains, and especially looks at memory. We hopefully will have this done over the next 2 years. We do recognize that this is not a randomized trial. It is a cross-sectional study, so we're not looking to prove causality from opioids. What we're looking for is just any kind of signal showing that the individuals with on chronic opioids, especially those that are on much higher doses than 90 MED for long periods of time, if there's some kind of signal showing that they do worse on these memory tests and have smaller hippocampal volumes. There are a lot of confounders, obviously, that can take place in this kind of study. We have somewhat limited it by limiting individuals to not taking chronic medications, which are known to affect cognitive function and may have or may cause some changes in the hippocampus as well. So things such as benzodiazepines, anti-epileptic medications, which are very common in chronic pain therapies. One of the other questions and possibly things that we can look at in the future are could opioid effects on the hippocampus be sporadic? As Dr. Kofke mentioned earlier on, not every single rat that was exposed to Remy Fentanyl or any of the other opioids suffered neurological insults. So that may open up some doorways to look at things like genetics. Does genetics play a part? And then ultimately, if we do show that there is some kind of signal, set up some forms of randomized trials or observational, longitudinal observational studies in the future. So the last part I'll talk about is a supplement that we put in to the R21 with Dr. Childress, or will be putting in. And this has to do with specifically looking at fentanyl in opioid overdose patients at the VA. This is just a picture showing how the number of overdose deaths, especially over the last 4 years or so, has skyrocketed due to fentanyl. And here you can see that 81% of the overdose deaths, this is here in Philadelphia in 2020, were associated with fentanyl. And so what we plan on doing with the supplement is expanding that to pain population group. So the chronic pain population group with chronic lower back pain and the controls, and recruiting another 35 patients from the VA who have a history of opioid use disorder and opioid overdose, and having them undergo the same test. So basically the cognitive behavioral test or the cognitive testing and the MRIs. Of note, these individuals, most of them still are on methadone therapy. And a lot of these, or the typical dosing, tends to be around 100 milligrams a day of methadone, which is very, very high MEDs. So for example, in chronic pain, we typically try to avoid going higher than about 30 milligrams a day of morphine. And we hypothesize that fentanyl overdose survivors on long-term methadone therapy for opioid use disorder are going to have smaller hippocampal volume on MRI and perform more poorly on neuropsychological testing as well. So I'll end it there. I know we're right on time, and I guess we can open it up for questions.
you so much, Ignacio and Jed and Andrew and Lauren Aguirre for joining us here as well. I do wish we had a half an hour for questions instead of a few minutes, but please, people join in, and I won't jump in and take the first question because I know there are many people who have thoughts.
Thank you all so much for wonderful presentations. It's extremely interesting. Dr. Barash, I had a question for you because you mentioned that the patients that you saw had clinically abnormal, or not necessarily abnormal structural MRIs, but then later you saw volumetric differences in the hippocampus. So I was wondering if you still have the T1 scans and can do more basically a review of free surfer or VBM analyses on these patients to see if there are also volumetric changes rather than DTI changes.
Yeah, no, it's a good question. We did it. So we had one case. There was a case in California that we published where the person had actually, and this is actually the main subject in Lauren's book, but it was a case where he had actually had an MRI like shortly before the event, had an MRI at the time of the event, and then we wound up doing an MRI at UCSF and follow up. I think I can't remember. It's like 6 to 8 weeks afterwards, and they showed about a 10% hippocampal volume loss basically from baseline to the 6 to 8 week mark when he was re-imaged. So the ones that we saw at Leahy were kind of like observationally, it was kind of like, they kind of eyeballed it, and you can see when you look at it, they do look smaller, but we didn't do like detailed volumetric analysis. The reason that the California case was so interesting was because it had that imaging before the event, with the event, and afterwards, where you could say, and there was like, I can send the paper out if you want to, but they have like, really good kind of like 3-dimensional analysis of it, and it's like pretty striking. And he's had, he's had longer lasting deficits with that.
And maybe one other question, if I may, Anna Rose. We're about to actually study tau in the brain in people with opioid-use disorder, and then also associate a history of overdoses to see if that accumulates faster. Do you think, and this is purely based on post-mortem studies, there are about 4 of them out there now, that's people who most likely died of an opiate overdoses show accumulations of tau, also in the hippocampus. Mm-hmm. Do you think that may be a biological marker of the behavioral changes that you see?
So it could be. So this is the other thing. So we did there was an IRB at Mass General where they were doing tau PET on patients with CTE and kind of brain injury related stuff. I had gotten them to agree to do a pilot study scanning patients with this amnestic syndrome, and we've gotten one person. So it's extremely hard. There's like a limited number of people. Their follow up is not very good and all that stuff. But we did follow up on one person, and that was a person at 4 years out who still had some like milder deficits. And the scan had no evidence of tau. I don't know what that means exactly. I'm trying to see if we can get a younger patient, because my theory was, maybe they have, maybe like in somebody who has isolated damage to the hippocampus, you'll see isolated, deposition of tau there. But we didn't see anything. It doesn't mean it's not there. I think in some of those pathology cases, there were cases where there wasn't necessarily tau associated with with injury. So it's hard to know, but that's a super interesting study, because nobody has. I've been asking people to do that for a while. Nobody has studied tau in an opioid overdose, which I think just in general is a really interesting study. They're actually systematically excluded from these studies. Nobody really looks at patients who have like substance use disorders. They're usually like a lot of times they're excluded from tau PET studies. So I'd be curious to see how that goes and what you find out.
One of the things we thought we might do in the patients that we're recruiting just to get the structural MRI is, if some of them are qualified and interested, is to pop them over and do a tau scan at PET, because we have the capability here. So maybe one way or the other we'll be able to answer this with the project that Corinne's talking about, or adding in a tau to some of the people that are doing.
Yeah, that's an awesome project.
The prospective supplement, and we'll see if some of them are interested, able. One of the things that's been always interesting to me is whether or not something, as you say, that's less than this full-blown amnestic syndrome may be occurring even with therapeutic and much needed use of opioids for either pain or for opioid use disorder treatment. And it's a very, very important question. And I think if we can show that there's a relationship between dose over time, in terms of equivalence, morphine equivalence, or look at obviously history of opioid overdose with fentanyl implicated, we'll be able, if we can show that correlation, that'll give us, it's not a random assignment obviously to overdose, we'll never do that, but to be able to show something that's a strong relationship. And so I think that once we get a larger number of patients where we can actually do something with dose and exposure, and maybe Andrew would like to comment on that in terms of doses within your animal studies, Andrew.
Well, relating it to humans, well, I don't know what we'll see. There's chronicity, and then there's acuity, and but nonetheless, the animal stuff suggests that there's a possibility that you could have a unexpected dose-related effect, i.e., higher doses, less damage.
Yeah,
I think that higher doses you probably start to get hyper. At the middle doses, you get inhibition of the interneurons, so that the regulators of the circuits get blocked, so that you get seizures. But I think maybe at higher doses you get hyperpolarization of all neurons. I don't know, and so that the blocking of the interneurons becomes not important. It's just a hunch.
It's very interesting, though. It's why it'd be really useful to get a lot of cases, even ones that have had exposure to opioids but not necessarily frank overdose. So I think there's just a whole wonderful territory to explore here. And you guys are so understated in your presentation, but this is actually really big scientific news in terms of understanding mechanisms. Bad enough the lethality, but not such good news that even if you survive, you may have some effects that are very long lasting and that really don't help with recovery, that really could make things more difficult all the way around. So,
I would hope that if we find an effect that warrants a subsequent study, I would hope that you could do a panel of a functional genomic study, not a GWAS, but pick SNPs that are relevant to pathophysiologic pathways to get an idea as to what might be important. That's how I would design it.
sure.
Something like that. So,
Absolutely. Lauren, did you want to say hello to everybody and say that you have a book that describes this journey?
Well, hello, everybody. And yes, I think you saw the book cover, and it was just, I really appreciated all the time people took to speak with me and explain the science, and it's just a fascinating story. So I'm glad I was able to share it with a book reading audience. So thank you.
For those of you who haven't dipped into the book yet, I thought I was just going to dip into it for a sampler, and it was so compelling I ended up, as I said to her, gulping it down over a long summer weekend. It was just too good not to keep reading it. So it's a wonderful integration of a lot of information about memory in addition to the stars of the show that we have with us here today. So we're very grateful to you guys for the presentation and also for the opportunity to collaborate. Very excited about that. And congratulations. It sounds like you're ready to almost enroll your first couple of folks. That's really good. And NIDA willing will have a supplement.
There you go.
All right. Any other thoughts? I know it's a little bit after 4. If not, thank you all. And I could have spent a half an hour just on the credentials of each one of these folks, but we plunged into the story to make sure we gave time for that. But I'm happy to let you know more about each one of them if you're interested. Thank you, everybody.
Thanks.
Thank you.
Really fun. Thank you so much. We've looked forward to
Okay,
this for a
see
long
you.
time.
Yep.
Take care.
Bye.